Thank you, Professor Mehra, for talking to us. Firstly, what I would want to ask you is about your childhood. And when you were growing up, India was also growing up at that time. So, how you saw India as a scientific nation?
Well, I was born in 1949, so just about 2 years after the independence.
Salman Rushdie's Midnight generation.
And my childhood was and I was born in Amritsar, and my childhood was in the 50s and 60s in Amritsar. I was playing around and all that. And we used to hear lots of stories about the partition at that time that had happened. One of the things that struck me when I grew up and started to understand things around me was there were many areas that were burnt around or they were demolished. And these were probably the Muslim colonies that were demolished as a part of the partition. And same thing had happened in Lahore. So during my childhood, we used to hear a lot about Lahore and Pakistan and things of that sort. So I studied in a very simple school, very near to my house. I grew up in a street like any other child of my age at that time would be. I belonged to a rather lower middle class family. And my schooling was all in Amritsar. From the pre-school, I went to the high school, and I had to walk about 4 or 5 miles to go to my school. And on the way, I used to see a statue of the Queen. So that used to be called as the Malka Buth. And that statue is no longer there now. But I remember I used to pass through that and all that, go over there. My brothers were more keen in plays and all that. They used to spend their time more in plays. I used to study all the time, and I remember I stood first in class 8 in my district, and they gave me a scholarship of 6 rupees a month. And when I went to class 9, so we were having school up to class 11. So 9th, 10th and 11th, 3 years of my fellowship, and that 72 rupees a year into 3, that was given to me in one go. That was huge amount of money at that time. And I was on top of the world. So, and my inclination was to go to medicine, biology, although my very close friends, they wanted to go to engineering, and they thought that I'm a mad guy because I had to dissect the frog and all that. But I did that. I therefore went to the biology side, and then from there to the medical college in Amritsar. And then I had never dreamt about that I would ever go to an institution like the All India Institute of Medical Sciences. So, life just takes a turn. I got admission in post-graduation in anatomy at AIIMS in New Delhi. And that's where I came in 1969. 1970. It was something very new to me, coming from a small town like Amritsar. And our life over there used to be very, limited to a few areas only. And come to AIIMS and you meet people from all over India. That was something really unique happening at that time. I finished my PhD in 1975. And my subject was to understand immunological aspects of leprosy or the mycobacterial infections. I was struck at that time when I went to the place called Sevagram near Varda. And the place is very famous, as you know, where Mahatma Gandhi spent 10 years of his life over there. And I also trying to nurture patients of leprosy, because leprosy in that area used to be very high. Mind it, in the early 70s, there were 10 to 12 million patients of leprosy all over the world, of which 5 to 6 million were in India. And this area, around Wardha area and Sevagram area, there was not a household where there was no case of leprosy. So I also started working on looking at the genetic aspects of leprosy, because we noticed that in the family, only a few children were affected, others remained healthy. So the idea was, and that was the time when the understanding about HLA genes was beginning to rise. And we were trying to understand the role of what we now call immune response genes. Are there immune response genes which in a way affect an individual, that some people go on to develop leprosy and others don't? So that was the whole understanding. Of course, I did those studies with the minimum of tools that I could lay my hands on. This was a very new subject in medicine and biology, and nobody understood in India because nobody had heard about it. But I don't know how I got fascinated with it, and I wrote to the NIH in the US, the National Institute of Health, serum bank, to give me some sera. We were having this serology-based test at that time, and they were very kind enough to send me the sera, and I did some studies based in Wardha. And that paper, my first paper, got published in 1975, and I was really on top of the world in an international journal. And this took me to the next step. And that's where my collaboration with the French started. In 1976, June 1976, Professor Jean Dancey, he was in a hospital Saint-Louis in Paris. He organized the first HLA and disease symposium in Paris. This was 4 years prior to he got the Nobel Prize, because you remember that he got the Nobel Prize in 1980. And he shared it with two other guys. And he was the only one who was working in the human system. The other two guys who got the Nobel Prize, they were working in the mouse system. And Dancey had maybe seen my paper, and he must have been fascinated by a young man who's trying to really show his face up in the third word. So he invited me. He sort of wrote a letter to me, why don't you attend the symposium? And I don't know from where I organized the money for travel and all that anyway. I talked to you about my activities in the 70s decade. And if I want to summarize that, the 70s decade became a very important decade in my life, because I had no idea that I will take up histocompatibility and transplant immunology as my subject for life. Actually, it went on to stay with me, because this subject was very new. Nobody knew at that time the importance of this. We were actually discovering HLA genes primarily as a tool to select a donor, a suitable donor for organ and bone marrow transplantation. But what we learned on the way was that this has a very important role otherwise also in a number of diseases. And we were in fact the first ones to start studies on HLA and infectious diseases. I started, as I told you, with a Dutch group on leprosy in the late 70s. And then I took it up even more in other diseases like tuberculosis and HIV there. And even today, a lot of people recognize me as a person with contributions, significant contributions on the immune response gene role, or the HLA-linked genes role in infectious diseases. So by the time the 70s decade ended, it was clear that I am now moving into this particular area. There was no department like this in AIIMS. There was nothing like this in AIIMS. But the administration, particularly with Professor Ramalinga Swamy, who was our director, he must have realized the importance of this subject. So he created a special faculty job for me, and that was in the Department of Anatomy. He created a job for me called the lecturer in HLA lab. and the HLA lab was situated in the Department of Anatomy. So that's how I came into the faculty of AIIMS. That was on 5th May 1979 when I joined as a lecturer in AIIMS, and with the task of establishing HLA services. And eventually, of course, this became a department of transplant immunology and immunogenetics, which even now stays over there. So I actually feel very proud and happy that I founded the first very department of transplant immunology and immunogenetics in the country and in AIIMS. But the 80s decade became even more exciting. That 80s decade took me to Australia. 80s decade took me to Japan, to Germany, to many other countries actually. And I was collaborating with my colleagues in Australia, particularly Roger Dawkins' group. He was a very big scientist in Perth in Australia, and he again during the early years that I met him in 1979. He helped me a lot. He helped me in how should I analyze my data, how should I conduct my studies, because my data, as I told you, that HLA studies, HLA was a new subject in India. There was nobody else other than me, so I had to really seek help from everybody, every corner. And so the Australian group, the Japanese group, the German groups, they all came forward, and I established very long-term, very fruitful collaborations with them, and I enjoyed working with them. In 1986, the Department of Biotechnology, Government of India, at that time it was called NBTP, National Biotechnology Board. So they created a research associate scheme. So I became the very first research associate of NBTP, and now called the Department of Biotechnology, to go and spend about 18 months in Seattle, USA in 1986. As a research associate sponsored by the NBTP Government of India. And Seattle was a wonderful center. This was the Fred Hutchinson Cancer Research Center, world famous for its contributions in the field of bone marrow transplantation. Again, Donald Thomas, who eventually got the Nobel Prize for his studies on bone marrow transplantation, he was the head of that unit in Fred Hutchinson. Of course, I went to work with John Hansen, but it was with the approval of Don Thomas. And Don, of course, also got the Nobel Prize, I think, much later. But he was very kind again. And this is where I first time learned the molecular techniques. The whole world was at that time learning to do HLA typing by the molecular tools. At that time, it was called RFLP. So that 86 to 88, that period was also very helpful to me, instead of bringing me to the next stage of bringing technologies back home of molecular tools. I had a lot of offers to stay back in the US and not go back to India. But anyway, I decided to come back to India. I came back to India in early 1988 and established all of those technologies over there. And by that time, I had also moved the application to the AIIMS administration to create a separate department of transplant immunology and immunogenetics. And of course, went through a lot of struggle. It was not that easy to create a sort of new specialty in AIIMS, but eventually it happened, and I became the professor. One of the very younger ones. I became at a much younger age. In 1993, I must have been only about 43 years of age. And then I became the head of the department. The new department was established first as a unit and then, of course, as a full department. And the department still continues and goes and does very good work. So the 80s decade was again a very prominent decade because the molecular methods of HLA typing or the PCR technology had just begun. You may know that the person who got the Nobel Prize for the polymerase chain reaction was, again, the work was done in 1986. So we were right. My group was doing work at par with the world, so much so that the international histocompatibility workshops, who are the highest international body, they included me in their council. That's why I became their counselor. Even till today, I'm the only counselor from the whole of South Asia, or maybe Southeast Asia. And so I think this was taking me to the next step. 90s decade was again a very prominent decade. I would say that most of the very productive developments in science that took place, medical science that took place, was in the 90s decade, because the human genome project was started in the 90s decade, and that really revolutionized the whole area of medicine and biology. I don't think there has been more significant impact than that of the human genome on the way we were doing now research in medicine and biology. And of course, by the turn of the century, by the year 2000, the publication came on the structural genomics of the human genome, and this was splashed in the journal Science, in the journal Nature, and then started what we call the post-genomics era. The foundations for genomics, genomic science, or the impact of genomics in almost every field of medicine and biology started in the 90s decade. And I think it was a very bouncy decade and great one, and we learned so much. But we were still a little handicapped because our access to good journals in time or discussion groups for us was far lower than my counterparts in the West, because mind it, I was competing with the best in the world, because there was nobody at the time, even at that time, there was nobody else working in India in the field of HLA. There were some pockets now coming up in, for example, CMC Vivellore, in Tata Memorial Hospital in Mumbai. But then the range of work, the range of activities that we were doing in AIIMS was much larger there. So our work then went on from autoimmune diseases to rheumatic diseases actually, and but more so now we were shifting towards better tools of selection of donor for organ transplantation and bone marrow transplantation. So these were very, I really recall those years were really hard years, the difficult years. But then so much to learn. The knowledge was being poured in almost every day at that time. The post-genomics era from 2001 to 2010, again, this was a decade when I must have done several training courses in immunology, several training courses in the field of HLA, and developed a scientific collaboration with many more groups around the world. I must state again that I had lost touch with this French group from 1981 to 1990, but then again an opportunity came in 1991. I organized the first HLA disease symposium in Delhi, in India, the Indian HLA disease symposium in 1991, and Dominic Charon came. And I've shown you the pictures of Dominic Charon. Dominic was the successor to Jean Dosset at Hôpital Saint-Louis in Paris. So we developed a great collaboration with his group. And Dominic has since then remained a good friend for several years. And then, of course, over the years, during the 90s decade and during the post-genomics era, I developed a lot of scientific collaborations with other very prominent French scientific groups, the most prominent of which is the Christian Boita and Jean-François Bach, Sophie Keller-Zuckman, all these people with whom we started working on in a collaborative way to understand the immunogenetics of type 1 diabetes. And we had several papers on that, and we were able to publish that the haplotypes, the HLA haplotype, the extended genotype of HLA that confers susceptibility to type 1 diabetes in India is very different from the West. And those are several papers that came out. Again, in 90s decade was again a very prominent decade because that time the Indo-French collaboration was at its peak. And from the Indian side, it was the ICMR, the Indian Council of Medical Research, and from the French side, it was INSERM. So ICMR INSERM would do every year a meeting, a joint sort of meeting of the experts from both sides, and try and develop, try and find areas where the Indians and the French scientists could work together. And I think this is where that I played a very prominent role. Again, I do remember having gone to several of these ICMR INSERM collaborative meetings actually in the 90s and in 2000, and I think the latest one that I went was in 2006. Again, a very important one, and I've got very fond memories of that, where the French side and the Indian side sat together and brought about a number of areas where we could collaborate. This continued even more, and that went on to until 2013-14, when we developed scientific collaborations with the group of Professor Christian Boita, a very important scientist in France, and we learned a lot from that collaboration there. And I myself visited France I think I must have visited France at least 10 to 12 times during that period of time. These collaborations, Indo-French collaborations, were very unique in one way. The uniqueness was that this was being both sides collaboration, that ICMR on the Indian side and INSERM on the French side, that when the project gets approved, it has to be approved from both sides first. And then there are visits planned, visits for the younger scientists to go over there and learn new technologies and maybe bring back to India. And then the principal investigators would also meet. So we had a number of opportunities to interact where my own students or young associates would go and work in French labs for about 4 to 8 weeks or maybe more, and collaborate with them and taking our own samples from India. And I think that really helped in advancement of knowledge on both sides. It was very, very, very useful. So these have been my really, the collaborations over there. And I must also mention here one other group in Normandy, Olivier Boy's group in Rouen. Again, we developed collaboration with them. Olivier and I were both council members of the International Union of Immunological Societies, and there were some other French scientists also. So there were more than one opportunity for me to continue collaboration with my French colleagues. And I'm very grateful to the government of France that they ultimately selected me for the Cavalier of the National Order of Merit in 2003, which I enjoyed very much. The function was held in the French Embassy in New Delhi, and the Honorable Ambassador of France actually gave me that prize on behalf of the President of France, and that has been one of the very high landmarks of my scientific career.
It has been a wonderful journey. We met at AIIMS.
Oh, okay. So yours was a love marriage.
Yeah, yeah.
Oh, interesting.
Means I liked him. After finishing my course at AIIMS, I went to Srinagar. I became a faculty member in the medical college there. So my home was just 2 kilometers away from medical college. It was very nice, and my elder brother was already an assistant professor in medicine there. So I had a good sort of life there also. And those days only this Sher-e-Kashmir Institute had come up. So why I had gone back was because I wanted to live with my family for some time. I knew after that if I'm to get married, I will be away and all that. So then my parents, initially they had hesitation. I didn't have my father. I lost my father at very young age. But my mother and my brothers, it was difficult because it was another caste, but still they were very broad-minded, and it has been very nice. He's basically a very honest and sincere person.
You know, in the 1971, I joined as a PhD student in AIIMS. And the subject that I chose was to understand the immunology of leprosy. And this we were doing by using an experimental model. And this was in the mouse. So we were inducing leprosy in the mouse. Because leprosy was a very popular subject at that time, a very important subject, because anybody who wants to learn immunology, he must work on leprosy, because the disease shows a very wide spectrum, and the immunological spectrum, which is really very fascinating. During that time, I also happened to visit Wardha area of Maharashtra, which is near Nagpur, and the the area near the Sevagram. Sevagram has a very good medical college over there. And the Sevagram is the place where you remember that about 10 years Gandhiji stayed over there. And Gandhiji was also was staying. I know my belief is that he wanted to let people know that leprosy is not contagious. So it is not that you should abandon those people. So he was staying in that area. That area was really very, very heavily infected with leprosy, and every family that we go, we used to find that. So I was fascinated with that area when we were moving around in the villages there, in the rural side. There were families with large families with 8 or 9 children, or 8 or 9 members in the family, all living in the same nutritional and environmental conditions there, but only 2 or 3 children would develop leprosy, others not. So that raised the question in our mind about, is there a role of genetics, or is there a role of what we call immune response genes in controlling the development of leprosy in people who get exposed to the M. leprae. And this is where then we started to work on HLA typing. HLA was beginning to be discovered as a tool primarily for transplantation, organ and bone marrow transplantation. But because of the work of 1972 of Bairuj Benasraf, where he discovered the immune response genes, and it became clear that the HLA system is required for presentation of any foreign antigens to the immune system. So therefore, it was imperative for us to study HLA genes. Of course, the technology at that time was very minor. We had these serology-based tools, and I must thank the Serum Bank of the National Institutes of Health USA for giving us some sera for free. And I don't know from where we borrowed or stole the micro syringe, and we started doing HLA typing work. But I didn't have the microscope. I didn't have the inverted phase contrast microscope, which is so very necessarily required. So I could do my work only through a monocular microscope. and I had to flick the tray, and many times the dye would actually get splashed onto my aprons also. Anyway, that study was done in 1974 and published in 1975 in this journal called Tissue Antigens. Tissue Antigens is now called HLA as a journal, but that's the top journal in this particular field. And I was very glad my paper appeared in volume 5 of May 1975. And that was a very proud moment for me. In 1976, Jan Dosse, who was the head of the laboratory in Hôpital Saint-Louis in Paris, and who eventually, of course, in 1980 went on to get the Nobel Prize for this particular field because he was the most important scientist at that time. He organized the first international HLA and disease symposium in Paris in June 1976. I was very keen to go over there. Dosse must have seen my paper, which appeared in 1975, and I did get a letter from him asking me to attend the symposium. And therefore, I don't know from where I got the money or whatever for my travel and all, and I did go over there. And that was a landmark in my career. Traveled to Paris in June 1976. I remember I had very little money. I must have stayed in the cheapest of hotels near Gare du Nord, and the symposium and the meeting was held in Palais de Congrès. So went by the metro or somehow I went over there. The first time I saw that huge conference hall, which I had never seen in India until then. But what was more remarkable at that time was I could see all of those people there who were big names in HLA at that time and whose names I had only read in books or in journals. All of them were there. And Dosse, of course, was a star. He was the organizing secretary for this symposium, and it was a real academic feast for us. So my paper on HLA leprosy was put up as a poster, and I was visited by a Dutch scientist. He asked me, is this study done by you in Delhi? I said, yes. In India? I said, yes. He said, my boss, Professor Jan van Rood, would like to come and talk to you tomorrow, so be here. Jan van Rood was a competitor of Jan Dosse, because at that time there was a lot of hot discussion going on. One of the two will eventually get the Nobel Prize, but the more votes were going more in favor of Dosse, because his paper had appeared in the scientific journal 6 months earlier than that of Jan van Rood. Anyway, next day Jan van Rood comes to my poster site, and I was very scared of talking to him. He was too big a man for me. So he of course made me very comfortable, saying, oh, is this study done by you? Very nice study. Done in India? Very nice. So you gather some courage, and then I wanted to tell him all the truth. I said, look, Jan, I have done this study, but I don't know whether what I've done is correct or not, because there's nobody to train me. And I didn't have a microscope, and I have done this study by so-and-so tool. I didn't have those fancy microscope at the time. So he said, would you like to collaborate with us? And that was like a bolt from the blue. I never imagined that a person of his caliber will tell me to collaborate with me. I was in fact looking for a center where I could go and do some postdoc work. So he then took me to Holland from there. Rather than returning to Delhi in 2 days' time, I landed up in Holland in his lab. And the laboratory that I saw over there was really an eye-opener. I hadn't seen that kind of a lab until that time in my life. And then we planned for collaborative studies. Jan and his team traveled to India in 1976 and in 1977. And for these 2 years, we had wonderful visits to Wardha, where we went to stay there for 4 to 6 weeks at a time, collecting right kind of leprosy samples from families of leprosy, and then doing the HLA-based studies. So in the first sort of visits in 1976, we were simply collecting blood samples from leprosy families, transporting them from Wardha to Nagpur by train, and then from there by air to Delhi, and then from Delhi again the samples will go on to Holland, and where they would separate out the lymphocytes and freeze them, because I was to then go in March of 1977 to Leiden, Holland, and do the studies myself over there. So that was the plan at that time. But in 1977, the second visit that we organized, we had become a little bit more technologically advanced, that we didn't have to ship the blood samples every day. We could separate out the lymphocytes ourselves in that makeshift laboratory in the Gandhi Memorial Leprosy Foundation in Wardha. And I must say here that all the staff of the GMLF gave us all support and encouragement. That was very, very nice of them. And we could separate out lymphocytes right over there. So we used to get up, and during my second visit, I should tell you the two stories of my work in leprosy in Wardha. During the first visit, the Dutch colleague that Jan van Rood sent, his name was Dr. René de Vries. René and I would early morning get up and go to the villages, because we wanted to catch the leprosy families right early on the day, otherwise they go to the fields. And one day, you remember in 76 was the year when we had emergency. Emergency was declared in India. And René and I, of course, went and bought a khadi kurta and pajama, and we used to go on bicycles to those families. It just so happened that René looked very similar to Sanjay Gandhi. You know, his head, the baldness at his back. He looked exactly, and after wearing the kurta pajama, the white one, and going on a bicycle, people thought Sanjay Gandhi is coming. And I do remember that particular day when we were going with our kurta pajama on a bicycle, and people mistook René as Sanjay Gandhi. And they shut their doors on us, and we could not get any sample. So those were one of the instances that we encountered. Of course, after that, whenever we went to the villages over there, we will go in a suited booted with our ties on, so that they understand, that they know that we mean doing good scientific work, and they were very collaborative. During our second visit, when I said that we did not need to ship the blood samples as a whole every day, we could come to the GMLF laboratory in the Gandhi Memorial Leprosy Foundation and separate the lymphocytes ourselves and freeze them in the dry ice. The dry ice used to be shipped to us from Delhi every 3-4 days, you know. So those are hard days, difficult time, and used to be very hot during those days. And you know, we were staying in that guest house of Gandhi Memorial Leprosy Foundation, and this Dutch man could not probably survive on the vegetarian food that he had to take every day, or could not be without any alcoholic drink. So one afternoon, when we were working in the lab, I used to work on this side of the table, and he would work on the other side. I remember about 3:30 in the afternoon, I heard a thud sound, and I saw Willem right on the floor. He was unconscious, almost dead, and that was very, very surprising to me. I really lost my wits, that what will happen if this guy actually dies. So anyway, I put water on his face and shouted at him, and all other people came, and he anyway got revived. And I do remember that we were counseled by the people over there, our seniors of that hospital, why don't you take him to Nagpur? So with that evening, we went to Nagpur, and had a hefty meal, hefty non-vegetarian meal with a lot of beer to this guy, and then of course it charged him even more. So these are some of the instances, some of the landmarks that you do remember doing the study. But I must say that those two visits, the studies that we did, they were published in the top journal, the Journal of Infectious Diseases at that time, and for the first time demonstrated that there is an HLA-linked control of susceptibility to leprosy. The first time it showed that. And that, of course, encouraged me to do similar studies in other infections. So I use the same model of doing studies in TB, and later on, a little more expanded sort of studies in HIV infection.
I want to tell you a story, a little story, a very fascinating one, of the visit of Professor Yom Bindu to India, to Delhi in I think it was October 1976. I went to pick him up at the airport, and on the way he noticed on the walls the swastika sign, And swastika sign, as you know, is very sacred to us, and we use it for every Hindu function, the weddings and anything else. But this was a Nazi sign for him. So he next day asked me that I have noticed everywhere with this sign everywhere. Is it not a hostility towards Europeans? I said, oh, maybe that you do not know. This is a sacred sign for us, and it is a little different from what the Nazis wrote, because here, as you see, like in Sanskrit, we have the little turning of the letter arm on all the four sides, and this is and we put the dot in between that. This is a sacred symbol for us Hindus. In every Hindu ritual, wedding, everything is used. So he was really thrilled to know that. Otherwise, he was getting a little bit off at this sign, because he said, you know that in Europe we hate this sign. I said, but this is different sign than that. This is swastika sign. The other, I remember during his visit when he went with me to Sevagram, and we went to Gandhi Ashram, and he was so fascinated by Gandhi. I think he was a follower of Gandhi. He sat down on that in that ashram and spun the charkha himself. And he spent a huge amount of time in front of the photograph of Gandhiji, particularly the photograph where Gandhiji is nursing Parchure Shastri. Parchure Shastri, as you know, was one of the patients of leprosy, and Gandhi himself used to nurture him every day and let people know that leprosy is not contagious.
If I were to ask you, what is that one thing that you would tell, say, people from my generation, that one that you should always do? What is that one thing in life that you would tell that this is something that everybody must do? What would that be?
That one thing. You see, what I felt is when I came to AIIMS, I became very thankful to even the school where there was nothing, but still thankful to my parents that they gave me a chance to go to school, because those days all girls were not getting the chance. And then to my teachers, who were whatever school. Then I went for some time to US from AIIMS on a sort of fellowship type, and then I became all the more thankful to the country that I come from, to AIIMS, that I had established some techniques there, which you won't believe. When I went to US in Washington, Seattle, Washington, University of Washington Medical School, there I established the technique of immunohistochemistry in that lab after going from here, because I had done it at AIIMS. And similarly, another advancement of technique called in situ hybridization, jo un dino, that DNA RNA things had just started coming out and probes were to be made, and we could localize the genetic materials, DNA, RNA, etc., inside the cell. And I did establish in their lab that immunohistochemistry I had already done here on by myself, and then they were very glad. They just wanted me to stay there. They offered me very good job opportunities, but my husband was bent upon coming back and establishing this subject here, so I had to come back. So what I meant by this is that you have to value everything wherever you are, experience. You go to school, however good or bad it is, it gives a base to you. It gives an opportunity to you for further rise. And similarly, every facility you get, you have to make use of it. You have to respect your teachers and feel free to discuss things like that. What I found there, it was initially hard for me to call them by first name, but she would just hammer me, you call me Anita, so I had to do that. So we have to sort of be interactive with our teachers, respect their knowledge, and sort of be productive all the time.